Hello, I'm Dr. Alan Yu. I'm Professor of Medicine and Director of the Division of Nephrology and Hypertension and the Kidney Institute at the University of Kansas Medical Center. I'm here to tell you today about a clinical trial called a randomized controlled pilot study of niacinamide in polycystic kidney disease. Niacinamide is a component of vitamin B3, and the overall goal is to test whether it's efficacious in slowing the growth of polycystic kidneys. Our plan is to enroll 36 patients. We're looking for volunteers that are between the ages of 18 to 60 with estimated GFRs of over 50. Patients will be required to come into KU for 4 visits over a period of 1 year. You'll have access to lab results, MRI scans of the kidney, and we can help with travel costs. If this sounds like something that you would be interested in, we are actively recruiting and need more patients. So please consider going to this website or calling the number on your screen to learn more. Thank you.
Dr. Barron has been the CSO of the PKD Foundation since February of this year, right? February? Okay. And he comes to us with over 20 years of experience in the pharmaceutical industry, taking on increasing levels of responsibility during this time. His most recent role was the Vice President of Non-Clinical Safety and Risk Evaluation for the United States and Europe for PKD Global Research and Development. So he is well equipped to talk about the sequence of drug development tonight. And with that, I will hand it over to David.
Innovation is what, for me, the drug industry is all about. It takes a lot of trial and error to bring a new drug into the marketplace. It takes a remarkably long amount of time. The sequence can be in years to a decade or more, and therefore it's a very high risk and high reward enterprise. And what I've learned over those 20 years is that many people are lucky to even see one drug go from the beginning all the way from its discovery to the marketplace. And if you've been lucky to see a couple, you still haven't seen them all. In fact, you haven't seen all that many, and each drug that's developed is unique. The development sequence, which is what we're talking about tonight, is pretty similar for each drug, primarily because the United States, the European Union, Japan, China, they all have regulatory authorities that have the force of law that tell you what you can and cannot do in developing a drug. So the sequence is pretty similar, but the issues that come up are pretty much unique for each drug. So just when you thought that there was nothing new under the sun, when you're developing a drug, there inevitably is something new under the sun. So R&D is a strong component. That's what I'm most familiar with. There's regulation, of course. I'm familiar with that because I have been before the FDA and EMA, which is the European Medicines Agency, many times. It's a very expensive endeavor for a lot of reasons, and I'll discuss some of those. And a lot of it is just plain luck. As I said, it's high risk and high reward, and I'll show you the statistics that really indicate how many molecules that are potential drugs never get developed. And even when drugs are marketed, they may never return the investment made in them by the company. They may not have enough patent life, they may not sell as well as predicted, or medicine may have advanced. Another company may have a better drug. So even though they get approval, they may never return all the investment that went into them. Now, I'm a toxicologist in part, and Paracelsus from many centuries ago made the famous quip that all substances are poisons. It's just a matter of dose. So many drugs, the issue even when they get to the marketplace is they may not get the right dose because there might be a 100-pound woman and a 300-pound man, or someone who metabolizes a drug differently than someone else, yet they market, by and large, single dose strength or several dose strengths, but it might be too high for some people and too low for others. And those drugs don't have a good uptake in the market. And also, we do controlled clinical trials, just like Dr. Yu's trial of vitamin B3. These are very controlled, and you're seen by a doctor on a regular schedule, but the real world is not a clinical trial. People forget their appointments, they don't take their drug on schedule, and so on and so forth. So there are many hurdles, and many of them are self-imposed by the companies themselves. So where does it all begin? It's really in basic science, and it's testing hypotheses. And when I say because with an exclamation point, is we have motivated scientists at drug companies who are scientists first and drug industry employees second. And they're just very anxious to test important hypotheses. So it's science for its own sake. But fortunately, good biomedical research always has value, and it's likely to lead to something that's important, not only for science, but also for the particular company that that scientist is working in. And despite all the hypotheses, many drugs are discovered by pure serendipity. There are many famous examples of this in other industries, like Teflon and so forth, that nobody was looking for a stick-free frying pan. It just happened that way. And we try to do rational drug design, meaning that we have a notion of what a drug should look like, and I'll talk a little bit more about that later. And then there are the medicinal chemists, and when I joined the drug industry from academia, I thought basically there were a lot of chemists and a lot of salespeople, but medicinal chemistry is just the beginning. And they make a lot of chemicals. And so we need a method of screening those chemicals for some sort of activity, and that's called high throughput screening. And this may mean thousands upon thousands of chemicals. And one or maybe none of those will ever see the light of day in terms of being a drug. And then there's the regulatory map, and I'll show you an example of this. And that's the real road to approval. You can't do this in a vacuum. You have to do this with the active engagement of the FDA or EMA or other regulatory agencies. And this really controls, in some sense, the pace of discovery. And then there's internal advocacy. Can you convince your company that your drug and your idea is worth their investment in time and effort? And that's also where troubles sometimes begin, because scientists are passionate about their discoveries. And when people like me, a toxicologist, says, boy, it works great in the test tube, but I'm not sure we'd want to give it to living organisms, that internal friction can really get high at times. So I was thinking, looking at this, that it really reminds me of the kindergarten game Chutes and Ladders and traffic circles. You're really trying to get from that idea represented by the light bulb, and at first you look at design and synthesis and screening. We talked about that in the previous slide. You pick out the best of those chemicals, and then you have to figure out whether you can make a dose form that actually can be swallowed or injected. Many times a drug that really has a lot of promise can't even be made into a dose form, and reluctantly you've got to throw that away, give it back to the medicinal chemists, and tell them to try to design something that can be made into something that an organism can actually be administered. And then, if you do have a dosage form that works, you go to animals, and that's in the PKD tox. I'll explain these a little more, but you have to make sure that the drug gets into the animal. You have to make sure that it has activity or pharmacodynamics, and you have to evaluate its toxicity. And one might think that if you do a study and there's no toxicity, that everything is great and you can go on into human beings. Well, actually, the worst thing that can happen in these early animal studies is that you don't see toxicity, because then the FDA tells you to go back and find toxicity, because they want to know what to look for when this actually goes into a phase 1 clinical trial. And here you test your best hypothesis about what the dose should be. You divide that by 10 or even 100, so you make sure that you don't cause injury to your volunteers. These are often college kids. And if the drug doesn't really work as advertised, in other words, maybe you take it but you don't get any blood levels, then you have to start and you look at yet another drug. And so you go through this traffic loop back and forth until you get an adequate phase 1 clinical trial again, that shows you can go on to actually looking at patients, not just normal subjects, but actual patients for the disease you're trying to treat, like PKD. And hopefully you can find a dose that is both tolerable and safe and has some efficacy. And from that study, using a small number of people, you go on to a phase 3 clinical trial. And these are the pivotal trials where you see a lot more patients in bed than were in the phase 2 trial. And you really test against either standard of care or placebo whether your drug has the desired effect or not. And if you think it does, then becomes the application phase, where you send in a new drug application to the FDA and they review it. And they're supposed to take about 12 months for the review process. If it's an innovative new drug, they sometimes take less time. And then hopefully you get approval, and the people who actually make the money in the company, rather than spend it, the marketing people, put that drug on the market. And this process is actually simplified here. There are many more traffic circles, and there are often times when phase 3 doesn't work, when you've already invested hundreds of millions of dollars into your new drug, that you end up back in the lower traffic circle. So I talked about high risk. Well, a lot of the easy blockbuster drugs are no longer being developed. They're already over-the-counter drugs like ibuprofen and naproxen and so forth. So the targets that drug companies are looking at, like polycystic kidney disease, are complex disease targets. They're expensive to study. The drugs may have adverse reactions that are poorly understood. Many of these compounds have poor absorption, meaning you take the pill, but it doesn't get into your bloodstream, as I mentioned earlier. It doesn't show efficacy. Mice are not just little humans. And there may be issues about patents. So unfortunately, if a drug company loses its patent protection, it usually loses its interest in further development of a compound. Some drugs don't hit only their own target, they hit off targets, and those usually cause side effects, meaning that your drug is unsafe. And then there are the chemical issues. The drug might sit in your medicine cabinet and decompose, and that's why you should, if you have prescription drugs, you should look at the take-by date, because drugs don't last forever. And then there's the competition. Someone may come up with a drug that treats the same indication or disease and does it better. There are other drugs that are just impractical to make. Most times the actual chemical in a drug is fairly inexpensive, but many times it is so expensive that you cannot set a reasonable price for the drug. So the reward. The drug is efficacious. It's safe, but remembering Paracelsus, it may not be safe for everybody, and you have to determine which populations you might have to adjust the dose for. For example, people who don't have great renal function may need to take a lower dose. People without good liver function, which metabolizes most drugs, may have to take a lower dose. And then you have to establish the benefit risk appropriate to the indication meaning what disease you're treating. If it's a life-threatening disease, you put up with a little more risk. If it's not, you're more on the benefit side. Also good to have is ease of dosing. If you can take a drug orally rather than having it injected or having to go into a clinic for an infusion, that is obviously a good to have. It must have an appropriate duration of action. In other words, you can't be taking the medication every 2 hours, hopefully. Hopefully it's once a day. And it has to have minimal interactions with other drugs or your diet. Some drugs are best taken on an empty stomach. Some drugs are best taken just after a meal. Hopefully you have a drug where it doesn't really matter and it doesn't interact with other drugs. For example, the FDA regularly asks drug companies, does your drug change the efficacy of birth control pills, for example? And some drugs do, and unwanted pregnancies can result. So these are a lot of the little things that are not understood about the drug industry that all have to be figured out before you really have an effective marketed drug. And of course, you have to be able to make it easily, economically, and reproducibly, and that's a critical issue because each batch has to be the same as the previous batch. And that's often difficult with complicated new drugs. And you must, of course, have the regulatory support to market the drug, and then you try to expand the drug's penetration into the market by looking at additional indications for the drug. And finally, to satisfy the people that make the money in the company, you have to have a positive return on investment. So that's the reward. The idea, and I'm borrowing from General Electric in the upper right-hand corner, is really sometimes you have a compound that's great, that the chemists love it, it's easy to make, and you're in search of a disease that it might treat. Sometimes you have a disease that's in search of a compound, and some companies, some academic institutions can spend years and years searching for that one compound that can actually treat the disease in question. Now, I refer to the good old days, and this still applies, that drugs used to be in a lock and key paradigm. The lock was the target or the receptor in the body, and the key would be the drug that either locks the lock, in other words, to turn off an activity, or unlocks the lock to turn on an activity. And that's how many of the classic drugs work, like ibuprofen, for example. And then there are exclusivity. 20 years ago when I started, if a company came up with a new high blood pressure medication, let's say, they often could market it exclusively without competition for several years. These days, you're lucky if you have several weeks sometimes of exclusivity because companies are all working on similar molecules. And now you're seeing a fight about the biologics. These are not small drugs. These are proteins. And even though their patent life has expired, the FDA isn't convinced that generic companies can actually make the exact same biological molecule, which are called biosimilars. But that might be the topic of another webinar. And blockbusters are not like they used to be. You've heard about the high prices charged for hepatitis C drugs, but they are not for wide populations like for arthritis or high blood pressure. Increasingly, drugs are for specific populations, and they're called precision medications. So that's the present, and the future is, let's get specific now about treating PKD, and there are multiple approaches going on right now. I really think this is a renaissance time for PKD. There are the small molecules, and these drugs prevent cells from proliferating, which is one of the reasons that cysts grow. There are anti-secretories that prevent the cysts from filling up with fluid, which also contributes to their growth. And then there are protein modifiers, and these can have very specific activities within the cyst. Now, key to all these small molecules is that they actually get into the cyst, so that the medicinal chemists very complex and can make you very famous if you get it right, so that these molecules go primarily to the kidney and not to other places where you do need proliferation and you do need secretion. Those would be side effects. And then we get into the biologics and the genetic attacks. And so these are antibodies, which everybody has. They fight, keep us from getting bacterial infections and so forth. And we can arm these antibodies that are made in the laboratory with warheads that target particular molecules in the cysts that cause the proliferation and so forth. There are even ways to approach PKD that probably won't be in the clinic for quite a while, but have been demonstrated in animals, such as kidney cell transplants that can get into the body by injection and sail underneath the body's immune system and actually support the normal function of the kidney. And then there are drugs which are under the classification of biologics, I suppose, but they're called microRNAs, and I don't have time to go into explaining these, but you might see them if you read articles about what's going on in the pharmaceutical industry these days. And then perhaps most famous, and which will probably lead to a Nobel Prize in the next year or two, are the so-called CRISPR technologies. This comes from the immune system of bacteria, of all things. They fight their own viruses, and it's been found that this technology can actually modify genes to correct mutations. And so obviously, since PKD is a genetic disease, I think that CRISPR technology is going to play an increasing role in strategies to combat polycystic kidney disease. And as I mentioned before, you want these drugs to get into the kidney and specifically into the cysts. If they sail around the body elsewhere, they may cause side effects. So the more specific you can target them, the better the drug is going to be. So why is it so hard? Why are we still waiting for the first PKD-specific therapy in the U.S.? Well, most PKD strategies require chronic dosing. You want to dose while kidney function is still fairly normal, and you want to dose probably for one's lifetime. So these drugs have to be safe, and the FDA is concerned, how are we going to know whether the drug really works? In other words, are we going to have to wait 35 years if we start dosing in a 20-year-old with PKD to know if there's really been a benefit? And will we really know that you can take this drug for potentially decades and still have it be tolerable and safe? Well, I'm convinced that compounds that are being considered and developed right now will prevent or delay end-stage renal disease in PKD patients, and I think that we will find drugs that are tolerable over time. That's my optimism, but from what I see, I think that that is coming, and through negotiation and discussion with the regulatory agencies and patient-centered approaches that Congress is increasingly interested in, I think we will be able to address these regulatory concerns. And then there are the big pharma concerns. You're the pharmaceutical company. Will your patent or your IP protection evaporate before regulatory approval? Will the benefit risk indeed remain tolerable over time? Even though you won't know that for sure, you certainly would not want to knowingly market a drug that would decline in its tolerability over time. And will you be able to price it at a price that's both affordable for the patient and will provide a return on your investment? And obviously, on the patient side, will it be effective? Will it be tolerable? And will it be affordable? And these are all woven in to the fact that big pharma are all profit-making, usually shareholder-owned companies, and so all these concerns from the patient, from the company, from the regulators take part in whether a drug makes it to the market. So there's lots of questions, of course, And this is Catch-22 Clinical Trial Edition. And this is an issue that comes up with many drugs. Most companies are really loath to develop drugs for pregnant women because the consequences of a drug that's not safe in pregnancy is obviously catastrophic. And so why don't they just let us do more clinical trials? And so the regulator says it's to protect you from the untested drugs. So really, what do you do? And I think that this is in part what we're negotiating through discussions and advocacy with the FDA and with Congress, that these concerns need to be addressed in a positive way for the treatment of PKD. So the R in R&D. The search for the target involves the pharmacologists, the physicians who treat the patients, market forces. Is there big demand for a particular disease? PKD is not exactly an orphan disease. It's on the cusp. But I think from an economic perspective, this is a nice target, fortunately, for the pharmaceutical industry. So you have to look for the right chemistry. Is it going to be a small molecule? That's probably preferable. They're easy to make. They can be taken orally, and they're easy to reproduce. In other words, each batch looks like the next batch. Biotechnology, that's fine, but at the moment, most of these drugs have to be administered parenterally, either under the skin or into a vein. And then there are molecular genetics, and then this gets involved with actually manipulating the genes within a cell. This is going to involve big data, and that's being increasingly accumulated. I think this is more for the future. And so this involves high throughput screening to get the right compound, and then creative chemistry to optimize that compound. And then seemingly you've got the best ideas and it just doesn't work the way you think you do. I've been, unfortunately, in several programs where this cartoon isn't as funny as it might be to you at the moment. We had a sepsis drug that really worked well at animals but didn't do anything in human beings. It's hard to predict these things. So what is the typical research path? And this is really the sequence we're talking about. This stresses the preclinical and the non-clinical, and I'll get more to the clinical in just a bit. We've talked about the target selection. We've talked about lead discovery. We've talked about what the medicinal chemists do. They're really the heroes in figuring out how to make a molecule that not only works, but can get into the body and stay in the body for the appropriate amount of time without causing toxicity. And then you do in vitro studies, not always because you want to start in the petri dish, but because the medicinal chemists can only make small amounts of the new potential drug. There isn't even enough to study it in a mouse. If it works in vitro, meaning in glass, then you go in vivo, meaning in life, and you test it out in animals and disease models. And then you move on to the really expensive part of drug development, and that's the clinical trials. So I think I've gone over the chemistry and pharmacology enough. One point would be the last bullet, and this often doesn't happen in companies, is that for most of the money makers in drug companies, a good toxicologist is a mute toxicologist. If a toxicologist has something to say, he or she better be darn sure that they have to kill a program. And this is some of the internal battles that are fought with companies that can lead to delays in development. And then there are the pharmacokineticists, a big word, but this means these are the scientists that determine how long the molecule stays in your system and how it's eliminated, how it's metabolized, and so forth. The toxicology, and when I first started in the industry, toxicologists were on the development side. Now they're often on the research side so that the pharmacologists and the chemists don't fall in love with their compound too soon, only to have it thrown back by the toxicologist because it isn't safe. But here's the regulatory part of the issue. We have to determine whether a potential drug is genotoxic. In other words, does it damage DNA? We have to determine whether it damages the reproductive tract or causes toxicity to a fetus. We have to determine the maximum tolerated dose. We have to determine chronic toxicity, assuming it's a drug that's going to be administered for months or years. And we also have to determine its carcinogenicity. And these are very expensive studies that can kill a drug at the very end when it's been all the way through clinical trials, because these studies are done over the course of 2 years in mice and rats and are legal requirement of drug development in the United States and Europe, and Japan for that matter. So pharmacology versus toxicology, you want to come up with the so-called clean dose, one that's bioavailable. I've talked about that a lot, but without really saying the term. This means that you put it in your mouth and it actually gets into your bloodstream. The pharmacokinetics I just described. The pharmacodynamics are based on the pharmacokinetics. You expect to see some sort of activity, and this measures at what blood level you actually see that activity. And then there's metabolism. How do you break down the drug? Are there active metabolites? So a metabolite is what the liver or the kidney or the gut does to the drug to break it down so that it can be eliminated from the body. But often the metabolites that are created are active and they may bind to DNA, which is a bad thing because that may mean that they're carcinogenic. You have to know the metabolic route so that you know whether it's going to interfere with other drugs the patient is likely to be on for the disease that you're looking at. And then another problem is protein binding. Your blood contains a lot of albumin, a common protein in the blood, and many drugs really like to bind to albumin, but if they're 100% bound to albumin, they're likely not going to reach their target because they're so tightly bound. All of these things, in one form or another, can mean that you've got to go back to square one in the Chutes and Ladder cartoon. It's complicated. I'm not even going to start on this slide, partly because this isn't my area, but the FDA has experts in all of this, so they want to know about high solubility drugs, low solubility drugs, high permeability, low permeability, poorly metabolized, extensively metabolized, so on and so forth. And each of these boxes, and there are 16 here, have consequences for the regulators. And the things that you have to do to approve a drug, it depend in part on all these kind of complicated charts. And all of these tend to be unique for each drug. So everything you learned about the previous drug adds to your basic understanding of these parameters, but each drug is unique and requires a unique solution. So CMC, and this is probably the area that I'm least acquainted with, is chemistry, manufacturing, and controls, is that the FDA wants to be absolutely certain of the quality control of what you're giving to human beings. And they will come and make surprise visits to your factory to make sure there are no impurities, that the compound is stable under whether it's humid or hot, whether it's uniform. Drugs are not typically given by themselves. They have excipients, which are lactose. You've got to be sure that you don't have enough lactose in there that you get into lactose intolerability issues. Packaging is an issue. Can a child get into an otherwise very dangerous drug or not? Believe me, we've had packaging issues in my experience that have delayed the introduction of a new drug by 6 months or more. So the D in R&D. Proof of concept is critical, and we've talked about that in the rodents. And modeling based on bioavailability, pharmacokinetics, and metabolism, which I've touched upon, and I realize this is a lot to swallow in a talk like this. You can model whether what you see in animals is likely to work in humans. The same with toxicology. And a lot of this, especially experienced toxicologists, can just look at a chemical structure and predict what kind of toxicity might come out of it. And so we do preliminary in vivo toxicology, and this is the basis for going to the FDA for an investigative new drug application that we can dose the first human volunteer. And I mentioned regulatory. Well, there are the GLPs, the good laboratory practices, the GMPs, good manufacturing practices, and perhaps most important, the GCPs, or good clinical practices. And the FDA is on you on a frequent basis to make sure that all these Gs are faithfully and consistently used in the development of drugs. And hopefully all of this together leads you to phase 1, and that you send in your IND, and if the FDA doesn't comment in 30 days, you're free to start dosing. This is, I think, the only occasion where not hearing anything from the FDA is a good thing. Often, you're in normal subjects, but if it's a drug for cancer, for example, the drugs are often very toxic and they will go right into patients. And what phase 1 is really trying to do is get that pharmacokinetics data I was talking about. In other words, can this drug really be dosed once a day, or do blood levels fall off quickly and you've got to dose more frequently? But most important, and this is why you tend to do it in young, healthy individuals, what is the maximum tolerated dose? So you keep dose escalating from a dose you know that's going to be safe, although sometimes you're surprised, to a dose that, you know, if you go any higher, people are not going to feel so good. And this is probably the only time you're going to do this, but the FDA really wants to know what that is if you run into trouble later in development, or if patients overdose either purposely or by accident. And then you move to phase 2, and as I introduced this in the chutes and ladders slide, you're doing dose ranging in patients. And so you're not only getting pharmacokinetic data, but you're also getting pharmacodynamic data, because you're in patients now. So some aspect of their disease that's measurable hopefully is changing, and hopefully is changing at a dose that is efficacious but not causing toxicity. And if you can find that dose in phase 2, and many times you can't, and back down the chutes to the beginning, but if you do find that sweet spot, you go on to phase 3, which are the pivotal double-blind registration studies. And for PKD, this can be a bit of a problem because, as I mentioned, while we're not truly an orphan disease, there are still relatively few people that are available to participate in these studies, and it's hard to get enough people to satisfy the FDA enrolled in these studies, and that's why we at the Foundation encourage you to be aware of studies that you might participate in. And then phase 4, it doesn't begin, say you get through phase 3, the drug works, it's safe, you're given marketing approval, you continue to have regulatory obligations. You have to take in reports from doctors and patients of any side effects that weren't seen earlier in development. And then, of course, the marketing people want you to look at new indications. So to borrow an old phrase, it takes a village. Again, I mentioned when I first went from academia to industry, I thought there were the chemists and there were people that went to the doctor's offices. Well, it's a lot more than that, and I've covered a lot of what's on this slide, but I'll tell you, the person in the middle, the program project manager or team leader, has their hands full. So timelines. It takes 3 to 6 years and 5 to 10,000 compounds, if we're talking about small molecules, to even get to the IND stage. Excuse me. And at this point, you go into clinical trials, probably with 5 of the drugs, and you go into phase 1, and typically most companies will really pick one drug that they're going to take through the clinical phases, phase 1, phase 2, phase 3, and as you see, what happens is you go from 20 to 100 people in phase 1, 100 to 500 people in phase 2, 1,000 to 5,000 people in phase 3. That's a lot for a relatively rare disease, and this program alone can take 6 to 7 years. And then, if everything works out and you feel your studies are positive, you submit an NDA. That can take anywhere from half a year to 2 years. I said 12 months, but occasionally they ask for additional studies, or they want to have certain questions answered. And then the chemists in the industrial scale have to scale up to manufacturing without even knowing whether you're actually going to have an approval, but you've got to make that commitment so that once you have the approval, the drug can be marketed immediately. And then you go on to the indefinite, hopefully, phase, and the company is obligated to do post-marketing surveillance. And for example, when I was in the industry, I had to read every article on a particular drug that I was responsible for and report to the FDA on an annual basis what was, how it was studied and what the results were, and were there any safety indicators in those studies. And those studies typically are done in academia and not by the drug company itself. And so here are all the regulations. I mentioned the IND, where when you don't hear, you're free to go, but you have meetings on the ongoing submission. You have an end of phase 2 meeting. 2A means early studies in patients. End of phase 2 meetings means you're finished with phase 2 and you want to present your rationale for phase 3. Then you have to meet prior to the NDA to see if the data you have is even acceptable. And then you make your market submission. And this is either an NDA or it goes under different names in different countries. This is ICH guidelines, or the International Conference on Harmonization, so that a drug company in the United States doesn't have to do totally different things to get a drug registered in Europe, so that the EU, Japan, the United States, and globally, the guidelines are basically similar, but some companies still have their own quirks, like Switzerland is not part of the EU, and you have to make a separate submission. So you usually have a large regulatory department to deal with all these various submissions and review processes. So the costs, and this is probably an overestimate, but for some drugs it's an underestimate, that in the 70s, and part of this is due to inflation, but part of it is just the cost of regulatory issues and the complexity of new drugs, it can cost up to $2.5 billion to develop a new drug. And you can see that return on investment is part of what's driving drug costs these days. And part of the reason it's so expensive is that if you look at the number of drugs per billion US dollars in R&D spending, the industry is getting much less bang for its buck over time, between 1950 and 2010. And not much has changed. When thalidomide, unfortunately, caused problems in Europe and tightened regulations in the US, that didn't really change the slope of this decline. There was a blip when the first biotechnology drugs came on the market for rheumatoid arthritis and so forth. And also the FDA was paid by drug companies to accelerate the review of new drug applications, and that helped a bit. But we're still back on the same curve. So it's becoming much higher risk and lesser reward because less compounds are coming to the market. So what are the alternatives? Well, one of them is the so-called virtual companies, and these are starting up in academia now. So basic research is done in the NIH. This has always been the case. But now research that's done in academia, many medical schools have their own patent attorneys, and they form virtual companies. And so you see an academic nonprofit biotechnology incubator, for example. You see academic and NIH screening centers. NIH conducts its own studies for drugs of high national significance like cancer or HIV/AIDS. And then you develop these consortia of clinicians and researchers in academic centers. And many of these companies are then bought by big pharma to take them into the most expensive part of development, which are the clinical trials. And you, the consumer, can help. Only 1 in 4 patients take their drugs the way they were prescribed. I was shocked when I saw this statistic. And that's why going from controlled clinical trials, where they know you're taking the drug when you're supposed to, when you get out there in the real world, drugs sometimes fail, not because the drug was bad, but because for whatever reason the drug wasn't taken as it was supposed to be taken. And then the FDA gets reports of non-efficacy, and the company might be forced to withdraw from the market. So as patients, we need to be cognizant of the fact that prescriptions are for a reason, and please don't save money by cutting pills in half or taking them every other day, for example. The way they're prescribed is generally the way you're going to get the best bang for your buck. So the future for PKD therapies. I think multiple targets are within range. We discussed some of those. The regulatory hurdles persist. It's a chronic use drug for an endpoint that could be years and years in the future. And so we need surrogate markers. And I think it's exciting that through the funding of the foundation, that total kidney volume for PKD is now an approved biomarker, and that approval comes from the FDA. So I think we've made progress, and I think we're going to make further progress in surrogate biomarkers, so we don't have to wait 35 years to make sure that end-stage renal disease is delayed or avoided. We have to have a good cost-benefit analysis, and we also have to realize that we can tell the FDA that there will probably, with an effective drug treatment for PKD, there could be a significant reduction in the U.S. dialysis burden, which is paid for by Medicare, the only such indication that is for people under 65, which means billions in savings that could be better applied to the NIH rather than dialysis clinics for future research. So we want to see an increase of quality of life and ability to work. And if you're talking to a congressman or a senator, that means more taxpayers. Even though they want to lower taxes, they still want that revenue. So if you're not on dialysis and you're able to work, that's a great drug, and that's what I think we're all looking for in PKD. Maybe we'll have to use polypharmacy, use not just one drug, but several. That's the case for many indications, like high blood pressure. So the hope is to go after cures and not just treatments, and that may be way back in the beginning when I talked about some of the genetic manipulations that are now available, the CRISPR technology and so forth. So these are other education opportunities coming up, and I'll stop now. I realize I've probably talked too long, but I'd be pleased to answer any questions you might have.
Well, there are a few questions, so thanks for that. And take a breath and get a drink of water, and I will, we'll get started. We did have quite a few, well, we had several really good questions, and I have one myself. So we are going to get started. Someone is asking, there's several about the FDA. So someone has said the FDA has recently altered its approval process, providing fast track approval for some kidney devices. Are you aware of this? And then has this impacted the drug development process as well?
So fast track is actually not a new thing. I'm certainly aware of the wearable dialysis belt. It has gone back for some tweaking, but it's still on the fast track. And I think a promising drug for PKD would probably be put on the fast track as well, because the regulatory issues are so thorny. So I think there'd be good collaboration between the company and the FDA, and I do believe that a promising drug for PKD would be fast-tracked. Often you get the answer instead of 12 months, you get the answer in 6 months. This might mean that the company is obligated to do some studies after approval, but it's a big difference. It may not sound like a lot, but a half year in the drug development industry means the difference between a positive return and a negative return.
Okay. And then, okay, so there's another one, but we'll skip over that because it's a totally other topic. And now we've had a couple of questions come in, wondering if you can explain the concept of an orphan disease and an orphan drug. I think you kind of referenced that a little bit, if people are wanting to kind of expand upon that a bit.
Now, I apologize for that. Orphan drug is a drug meant for an orphan disease. An orphan disease, for example, autosomal recessive or AR-PKD is definitely an orphan disease. Cystic fibrosis is an orphan disease. These are often diseases that occur in maybe 20,000 individuals, but less typically. The limit in the definition of orphan disease is about 200,000 patients. There are some diseases for which there may only be 70 patients, and believe it or not, there are drug companies that are working to satisfy their needs as well. But there are thousands of orphan diseases for which there is no effective therapy.
Okay. And so, if you said this, I missed it. AR-PKD would be considered an orphan disease.
Definitely.
AD-PKD?
The AD-PKD is a curious, it's a little above the threshold, but it's still a relatively rare disease. Definitely well under 1 million people in the U.S., but probably more than 200,000. So the FDA has, in my opinion, taken a liberal approach to its designation. And most importantly, they realize that there's a significant population out there that has no specific therapy for the most common genetic renal, potentially fatal disease.
Okay. Okay, so we're getting a few more questions in now. So, okay, back to the idea of fast track status. Someone just kind of commented that fast track status is determined if there is an unmet medical need, meaning there's no current treatment for the disease. Is that an accurate comment?
Yes.
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 Okay.
But I would stress that fast track is the approval process, not necessarily the development process. So
it's very different. I mean, the development process is really what takes so much of the time.
Right.
Right. Okay. So kind of related to that, someone has commented, and this is sort of a comment on PKD Foundation policy, but not policy so much as behavior. But given that drug development is an arduous, expensive, and risky process, would it be better for PKD research, maybe not the foundation so much, just PKD research in general, to focus, excuse me, on research of behaviors that could slow the progression of PKD in individuals? So things like diet, exercise, water intake. Would we be better to focus on that as a research area rather than basic science or drug development? What's your opinion?
Well, I think the foundation does support research on diet modification and behavioral modification. We would certainly consider those types of grants. the basic science has to be done, and I'd like to talk about that more in the future, but that's where the next therapy is going to come from. The questioner is correct that we are not of the scale that we can support a complete drug development program, nor is that our mission. That's for the drug companies to do. But we can, however, provide for active clinical trials. For example, the trial going on at Indiana University looking at pioglitazone and PKD, and we can supply, for example, the compound and the placebo compound for those trials. And I think that is money well spent, because this could result in a quick new therapy, because the drug is already marketed for other indications.
So do you think diet studies and water intake studies, things like that, are well supported through entities other than the foundation? So NIH money or things like that?
So there's an organization called PCORI, which is actually a government-funded company that looks at pragmatic studies. And by pragmatic, it means you don't have to go in for any complex procedure. It is things like water intake or dietary change. That's one possibility if there were to be a group that would apply to PCORI for such a contract to study those kind of things. And the NIH does do large-scale studies involving diet and behaviors in terms of blood pressure and diabetes, for example.
Okay. So it's 8 o'clock, and we have 2 more questions that I want to ask. So I'll just say, if you're able to stick with us for a few extra minutes, that's great. If not, but you want to catch the end of this, it's being recorded, and we will we'll put this on the website. So if you want to catch the end of it after the fact, because we are going to run just maybe 5 minutes over, catch the end of it then. But one thing I want to ask, and you might have mentioned this and I just missed it, but the patent approval process that you mentioned, that one thing that drug companies would be concerned about is how long they have patent protection. So can you expand upon that a little bit?
So when the chemist first discovers a new drug or the new biological, there is a decision made at the company, when do we actually apply for the patent? Because once the patent application is out there, other companies know that you're working on something they're working on. And so it becomes a game of chicken where these 2 cars are driving head-on, and who's going to blink first? You don't want to patent your drug too soon, because the patent will be over by the time you get your approval, but you don't want to apply for the patent so late that someone beats you to it, and then you don't have the patent protection that you need. That's an interesting question, and a lot of effort in drug companies is devoted to patents. Often you can write a patent in such a way that it's not clear in a tree of different molecules which is the actual molecule that's your lead molecule. So there's a lot to that. Yes, indeed.
And so once you get patent approval, did you maybe just reiterate how long does that company retain that before the drug can then go to generic?
I believe it's 17 years. I'm not certain. The other thing, if you are really out of patent protection, but you're the first to bring that particular drug to the market, you will be given exclusivity by the FDA, typically for 5 years. Even though you've run out of your patent life, the generic drug companies cannot come in and copy your drug for 5 years, because the FDA does recognize that companies can't stay in business if they can't make money from their
all of the resources into that drug, so they get 5 years when no one else can sell that same
yes.
drug. Okay. Okay. So I think we have several questions, but I think we're just going to take the time to answer this one last one. Are there drugs in the pipeline that are already in use for other diseases. So I'm assuming they mean, are there drugs in the pipeline for PKD that are already used for other diseases? And I think we know that there are several, so maybe you could just mention them.
So oddly enough, niacinamide, or vitamin B3, is used sometimes in high blood pressure. I mentioned pioglitazone, that's also known as Actos. That was a diabetes drug. And oddly enough, another diabetes drug, metformin, which doesn't have the same mechanism of action as pioglitazone, is also in clinical trials for PKD. And there is a company called Cadman that has a drug in phase 2 for polycystic kidney disease. So that actually there are a fair number of programs that have made it to the clinic for PKD. So I'm rather excited about that, and I think that there'll be more in the new.
And there are several studies going on. You said that? There are several studies going on right now, clinical studies that patients could participate in.
Yes, some of these are rather local. If you're not in, for example, the pioglitazone study is really centered in Indianapolis. But if you're willing to travel, if you live in Bloomington or South Bend and you're willing to come in when it's required for the study, please do contact Indiana University.
And there's a study at the University of Kansas looking at B3 or niacinamide,
yeah,
which is kind of a different take on it. And then there's also tolvaptan is really.
Yeah, so tolvaptan, the reprise study, I've been told that that's how it's pronounced, not reprise.
I've been saying
So that's still enrolling, and that is a drug, tolvaptan is actually marketed in the United States for the treatment of congestive heart failure.
So certainly if you're looking for something that you can do to take part in this drug development issue we have, consider learning more about clinical studies, and if one fits, then look into potentially participating.
And I should add that tolvaptan is a study that you can probably enroll in at a site relatively near to you countrywide.
They're all over the place.
Yes.
And you can find more information about that on our website, pkdcure.org, and just look for the clinical study page, and you'll see all of the potential studies available in your area. Well, that is all the time we have. Can you reverse to the education? Yep. So be sure to check out these education opportunities available. We have the blog. So we have patient stories, education posts, stories. Sometimes we have posts from physicians and researchers. So really cool stuff. PKDconnection.org. It's a great place to stay connected to the latest and greatest that we have for you. PKD National Convention coming up in Orlando, June 24th through 26th, 2016. So it will be on Walt Disney property. So start gathering your pennies together and make a vacation out of it. pkdcure.org/convention for those updates. And then finally, our 2016 webinar schedule will be coming soon. We'll still be doing the webinar Wednesdays, every third Wednesday at noon with David to discuss the basic science of PKD, and then we will also be doing these evening webinars, several throughout the year. So if you have topics you'd like to see us discuss, please email me your ideas. And that is all we're going to do. We appreciate the opportunity to provide you the best possible education. We'll be emailing a quick survey in the next few days. We appreciate your feedback, and we'll use your responses as we develop additional resources and programs in the future. If you need to reach out to us directly, or if there's a question that you felt we did not answer or you want more information on, please just shoot us an email at education@pkdcure.org, and a member of our education team will get back to you. On behalf of everyone at the PKD Foundation, we wish you a lovely evening. Thank you so much for being here.